In our previous videos, we did a basic review of chemistry, things like the structure of the atom, covalent bonds, ionic bonds, properties of water. Now what we're going to finish our chemistry unit looking at is organic chemistry, or the study of carbon-based molecules. We study organic chemistry in biology because cells are consisting mainly of carbon-based compounds. Things like proteins, nucleic acids like DNA and RNA, carbohydrates, fats, and lipids. And that distinguishes living things from non-living things, or these organic compounds. Now, in organic chemistry, all that word organic means is that the compounds contain carbon. That's all that word means. Now, additionally, most organic compounds have hydrogen atoms too. So something like methane is going to have carbon and hydrogen, but carbon dioxide, CO2, does not have hydrogen, so it is not considered organic. So that's kind of our two major characteristics of organic compounds. They have carbon and they usually have hydrogen. Now, why is carbon so important and so crucial to living things? The main thing about carbon is that it has 4 valence electrons. Now remember from our review of the structure of the atom, all that word valence electrons mean is that it has 4 electrons in its outermost shell. What this translates to mean is that carbon can form up to 4 bonds in order to make itself stable and full of electrons, right? 4 plus 4 is going to make 8, which is our outermost electron shell stability number, if you want to call it that. So those 4 valence electrons, again, mean that carbon can form 4 bonds. Additionally, these bonds can cause the molecules to have chains or rings, and these bonds can also be either double or triple bonds. This tetravalence, this 4 valence electrons, makes carbon able to have huge, complex molecules. Here are some examples of some small molecules of carbon. Methane, made of 1 carbon. Ethane, made of 2 carbons. Ethene is also made of 2 carbons, but notice there's a double bond here that makes it different than ethane. So there's more hydrogen around these carbons than there are around these carbons, because this one has a double bond and this one does not. Now, as we said, carbon typically bonds quite often with hydrogen, but carbon can also partner with other atoms such as oxygen, nitrogen, and other carbons. Usually we see these 4 atoms in what we call the building code of organic chemistry. These 4 atoms are the ones that are the most often involved. For example, here's a molecule called urea. It sounds like urine, right? That's a molecule found in urine. Notice the building block in the center would be carbon, and attached to that carbon are nitrogen, oxygen, and then the hydrogen are attached to the nitrogen there. So that gives us a pretty big diversity of combinations that we can make with molecules with carbon, oxygen, hydrogen, nitrogen. Now, in addition to those partners, carbon has a lot of skeleton diversity. So carbon chains can vary from 2 carbons to hundreds and hundreds of carbons. We have ethane, which has 2, propane, which has 3. So we can build carbon chains that are huge when we're talking about organic molecules, and you will see those eventually when we look at something called hydrocarbons. In addition to length, carbon also has diversity because the carbons don't necessarily have to go in a straight line like they do here in butane. Instead, they can branch. So we don't have this nice straight line, we have this branching structure. We call this isobutane. Like we said before, the double bonds play a role in the variety and the variation of carbon structures. This is made up of the same number. These 2 molecules are made of the same number of carbons and hydrogens, but because of where the double bond is, here you can see it between the first carbon and the second carbon, and here we can see it between the second carbon and the third carbon. That gives these 2 molecules unique properties and makes them 2 completely different molecules, as you can see by their name, 1-butene and 2-butene. Lastly, our carbon is very different, or very unique, because it can form rings. So not only does it make those straight branching, or those straight chains, I'm sorry, but they also make the branching chains. Carbon can also make these rings. We're going to talk about a lot of different types of carbon rings where that straight structure folds over on itself and makes these carbon rings. This ring, cyclohexane, has single bonds between the carbons. Benzene, this particular ring, has double bonds between the carbons. So as you can see, even though we still have a very similar number of carbons here, they can make two totally different structures based on the single bonds or the double bonds. So that's kind of A, B, C, D. Those are the different ways that molecular diversity can arise by carbon. So we talked about different lengths, we talked about the different branching, we talked about single bonds versus double bonds, and we also talked about carbon forming rings. So those are the 4 different ways that carbon makes a very, very diverse number of molecules. The next thing that we're going to talk about are something called hydrocarbons. Hydrocarbons are organic molecules, as you can see. Hydro, that part meaning hydrogen, and carbon, obviously meaning carbon. Hydrocarbons are large molecules made up of many, many, many repeating carbon and hydrogens. As we can see, we're going to take a look at a picture here. This is a hydrocarbon. The black is the carbon, the gray is the hydrogen. This kind of is a smaller version of this molecule, but a typical hydrocarbon would be made up of repeating hundreds and hundreds of carbons bonded to hydrogens here. That is in a fat molecule. This whole molecule is called a triglyceride with 3 fatty acids or 3 hydrocarbon tails here. So we're going to see that hydrocarbons, usually in fats and other types of lipids, have tons of energy in them, and they undergo reactions that release a lot amount of energy. Those hydrocarbons, because they're just hydrogen and carbon bonded to each other, they can store a lot of energy inside of them. As we can see in this picture, fat molecule stored in fat droplets in adipose cells. Adipose cells are our fat cells in mammals. That's where we store excess calories. And we will spend lots more time looking at different types of molecules like hydrocarbons and fats. This was just a little preview to give you an idea of what a molecule that is organic would look like and its function. The next thing that we're going to talk about is something called an isomer. An isomer has the same formula, so the same number of carbons, the same number of hydrogens, the same number of oxygens, whatever, but they differ in their structure. So our first one, a structural isomer, best to just look at a picture. Structural isomers have the same number, again, so if we count up our carbons and hydrogens, they're going to be the same, but their shape is different. This goes back to that example of the straight chain versus the branching chain. Again, this is one molecular shape, this is a different molecular shape, and as we've talked before, molecular shape in chemical reactions and interactions between molecules is very, very important in biology. So this molecule would have a totally different function than this molecule. That's our structural isomer. Our second type, a geometric isomer, again, have the same number of atoms, but the key thing with this is there's a double bond involved. If we look, double bonds here in the center, but where the attachments, where the groups are located around that double bond differs between these two isomers. Usually the cis isomer, these two groups, let's say this was an OH group or a phosphate group, both of them are on the same side. That's a cis isomer. These X's, again, could be a phosphate, could be a hydroxyl group, could be a methyl group. They are on opposite sides of the double bond. We call that trans. So key thing about a geometric isomer, there's a double bond involved. Because double bonds are rigid, they do not spin, they do not rotate, these attachments, these other groups, will stay in that position. Okay, so that makes sense there. The last type of isomer, and this is the one that's the trickiest, is what we call an enantiomer. Enantiomers are mirror images of each other. Again, they're going to have the same number of atoms, the same structure, or the same molecular formula, but they're going to have a totally different structure. I will make a model of this for you in class so you can kind of get your hands on this and see this firsthand. but you can see that there's a carbon in the center, and there are 4 different groups attached to the carbon. How these groups are arranged around that carbon makes them totally different. This enantiomer and this enantiomer, a good way to think of these, they cannot physically be placed on top of each other where they will overlap. For example, if you take your left hand and your right hand and try to make them so that they are in the exact same place in space, that is physically impossible because they are mirror images of each other. Again, this one's a little hard to wrap your head around. We'll talk more about that in class when I see you guys next. Now, the reason that we talk about these is because enantiomers, again, structure matches function. The shapes of these are very, very important to the function of those molecules. And one example is in the pharmaceutical industry. If a drug is manufactured and there are 2 different enantiomers, those enantiomers may have different effects in the patient. Again, something very, very small, a change in the molecule could have a totally different reaction in the patient. So, for example, a drug that you've probably all taken before, ibuprofen, Aleve, that kind of stuff, which is used to treat pain, inflammation, headaches. There are 2 different enantiomers, the effective one and then the ineffective one. So we have to be careful when we're manufacturing these, that we're making or that we're distilling or we're, I'm trying to think of the word that I'm looking for, that we're extracting the correct enantiomer out of that product, because if we treat a patient with this, it's not going to work. Now, the last section of this chapter is going to go over the different functional groups, and you may have heard me say some of them as I was doing this lesson. Things like hydroxyl, carboxyl, I think I said methyl group, phosphate group. Those are different groups of atoms that are attached to the central organic molecule. So they're attached to the carbon somewhere. And there are a number of different functional groups that we're going to be talking about. Let's take a look at what these guys look like. So what you're looking at here are 2 hormones, estradiol and testosterone. 2 hormones that, because of 2 slight differences in their functional groups, will lead to the anatomical and physiological differences between a female and a male. This is the only thing that makes us different, basically. On the female hormone, there's a hydroxyl group. On the male hormone, there is a methyl group, and there's also a double bonded oxygen here. We will see that these different functional groups have different structures, again, which leads to different functions. There are 7 different functional groups. I'm not going to read these to you, but you are going to be responsible for knowing the structure and basic functions of them. So on your little note-taking guide, one of the last questions is to take a look at the 7 functional groups. You can find this either here in the PowerPoint, or also this goes into quite detail in your text. Again, just go through these, kind of know if I say carboxyl group, this is what you should be thinking of. If I say amine group, this is what you should be thinking of. So just kind of get yourselves familiar with these. Again, structure and function. Thank you.